everybody else for today, and let's welcome the other Orange County High School group.
Hello and welcome to the Orange County High School Block 2 Arabidopsis Research Presentation. I'm Griffin and this is Alden, Caroline, Corinne, and Joe. In this experiment, we were testing the effects of drought on the plant Arabidopsis thaliana. We used Arabidopsis because it is a good model organism as it has a small genome. We were also trying to find out the function of the AADL gene, which is the function of the production of flavonoids. Flavonoids are chemicals that in plants that aid their growth and development as well as their color. They also have effects on humans as they can help against cancer and other deadly diseases, so it is important for us. Our hypothesis was that in the experiment, the wild type plants which had the AADL gene would do better than the mutants which did not, because the mutants were missing a gene that aided in growth and development, so they would do worse when exposed to drought conditions. We tested drought because drought is something that occurs in Arabidopsis environment, so we should test that.
For our experiment, we tested the effects of an artificial drought on our plants to see if the AADL gene had anything to do with the plant's reaction to drought conditions. We conducted the experiment on 4 groups of plants, including 2 experimental groups and 2 control groups, each containing a pot of plants with the AADL gene and a pot of plants without the AADL gene, and each pot included 4 plants. We watered the control plants daily and watered the experimental plants when they showed signs of dehydration, like leaf decay or weight loss. We placed all of the plants under lights for 24 hours. And each group of students took turns watering the plants, and observations were recorded daily.
After our data was recorded and collected, it was placed into the graph above. Many trends in the data can be shown in this graph. One of the main trends is that the wild type control, which is the blue line, had the consistently the longest rosette width. Another trend in the data is that the wild types seem to go together and the mutants seem to go together, showing that the experimental didn't have that much effect on the plant growth. In the picture, it shows that the wild types have longer rosettes and the mutants do not. Those are both experimentals receiving drought conditions. Another thing about the picture is that it's shown that the wild type has a darker coloration than the mutant. This is due to the production of flavonoids. One part of the data in the graph that is shown is right here around day 15, where everything had been kind of going around the same rate until everything kind of comes together. And one point to point out is that the green line, which is the mutant experimental, and the red line, which is the mutant control, kind of flip-flop. So the wild type then grows faster than the mutant, which is different from what we've been seeing before. Another point of data to point out is on day 18, where the red line, which is the mutant control, kind of takes an unexpected turn up. And then the next day of data, it goes back down. We don't exactly know why this, believe it could be of human error. We measured in centimeters, but some people could have accidentally done millimeters. Another big thing is our light source. Our plants grew upward, some of them, and some people flattened out the rosettes where others didn't, so they could have measured a taller plant that had the same length. And then around day 18, again, there's another conjunction where it flips.
This is the phase 2 analysis graph. This is the overall class data for the average goal height. As you could see in the previous graph, phase 1 followed our hypothesis exactly, and in this graph, the mutant experimental is way up there, which is the opposite of what we expected. I don't know if you guys can all see this, but the green one is the mutant experimental, which is the one all the way at the top. The red is the mutant control, which is red. The yellow is the wild type experimental, and the blue is the wild type control. So starting off with the wild type control, that one started at the very bottom and made its way up to actually meet the wild type experimental, which they kind of stayed together until around day 28 and then separated, but they came back together. That just is because of the experimental element of drought. The green and the red, so the two mutants, the red one, which is mutant control, actually started off in front of the experimental mutant, and by the end came actually the lowest one. So that went from the tallest to the lowest. And the green mutant experimental started around the top end. Did it fall all the way up? Okay.
In conclusion, we can see that our first part of our hypothesis was correct as following phase 1, showing that the wild type was to do better than the mutants. But then once reaching phase 2, where we're measuring full type, we saw that the mutants, in turn, did better than the wild type. Now, the reasoning for this, we're not sure. One of our guesses is that the AADL gene had something to do with whether the plants could fully detect that they were even going through a drought. So they were putting all their nutrients and energy toward, and they can have, as a result of this, you can have sterile offspring. To test this, you take the seed pods from the Arabidopsis that were tested in this case and plant them to see if they were sterile or not. Special thanks to Mr. Lally for his citations for this project. I was referring to if the plant was putting forth all of its energy towards growing, and that it wouldn't have because it's lacking the AADL gene. If the AADL gene was evenly spreading out its nutrients and growth habits, it was putting all its energy towards growing. That's why it was behind at first, but then caught up quite rapidly.
So you're saying those plants were sterile, or that they created seeds
That they could create seeds that were
that would be sterile?
Yes.
Okay, so you said you were testing drought. How often were the drought plants watered if they were watered?
This is a good question, because the way that the drought was, when you have a drought, you can't have a watering schedule, because it is, but it's irregular. Because you can go to the point that the plant starts showing signs of drought, so stress such as curling of the leaves, they start turning yellow. But if you have a regular day, say we only watered once a week, if they were starting to dry up in the middle of the week, they would die and we wouldn't have plants to test. So we would go until they would get to the point of showing signs of stress, of drought, then they'd be watered.
How do you measure 27.5 days?
The honest answer would be a calculator.